This is lecture NS-2. In this lecture, we're going to start by talking about how action potentials are propagated down an axon, and we'll also get into the concept of saltatory conduction and myelination. We'll then talk about what happens when the action potential gets to the axon terminals, the concept of synaptic transmission, including neurotransmitters and receptors, and how that whole process works. We'll then get into the idea of senses, and we'll talk about what our senses are and the idea of sensory transduction, different types of receptors, and the idea of receptive fields. And then finally, we'll get into the specifics of one particular sense, just to talk about one example of how one sense is done, that being phototransduction, or the process of transducing light into nerve signals. All right, now you've learned about action potentials, one of the major parts of nerve signaling. Let's talk now in a little more detail about what happens to that action potential as it propagates down the axon. So, we'll just sketch in here an axon. So here's our axon hillock. And as we talked about these action potentials, you got the idea that when one part has an action potential, if that's its depolarization period, those positive ions, the sodium coming in through those voltage-gated sodium channels, spreads out from there. Really, this is starting another graded potential, or adding to the one that caused this action potential. And as those positive ions, we talked about the ones going backwards. Those are hitting areas that are in their absolute refractory period, so they don't really do much. But these ones going downstream bring this part up to threshold and start its action potential. And then as the ions come in, that brings the next part up to threshold and starts its action potential. Remember, an action potential technically is something that happens in a particular place. So this is not a spread exactly, but rather what we call propagation. Which really means more like reproduction. It's like this action potential causes one here, which causes one here, which causes one here. You can think of that as the action potential sort of pushing and re-energizing a graded potential and keeping it going down the axon. The analogy maybe being if you had a ripple in the pond, you could keep it going by having people toss pebbles in right behind the ripple, which kept adding to it, preventing it from dying out. This would be a little bit like that. Now, there is the interesting question of how quickly does this go? That has two aspects. One is that it depends on the size of the axon. The narrower the axon, the more effectively friction there is. That as these ions get pushed down, the more likely it is that they're going to effectively kind of bump their way into other ions and lose some of that momentum. That's not exactly the right way to say it, but it's sort of what I'm getting at. There's more resistance to current flow, which means it effectively takes longer to start each action potential, so the signal doesn't propagate as quickly. The narrower the axon, the slower it goes. When people were first studying these action potentials, they first figured out how they worked by using what we call squid giant axons. These are not from giant squid, although giant squid are very interesting. But instead, they are from regular squid, which have a few axons which are enormous. There are axons in some squid that are 1 millimeter across. That's a cell with an axon big enough to see with the naked eye. That's pretty impressive. So that made it possible to actually get tiny electrodes onto those axons and study what was going on that cell membrane. Now, that allows for very, very fast signal propagation. In an axon that big, this signal can propagate down the axon very fast. And in squid, that allows them to have very, very fast reactions to some kinds of stimuli. Humans also would like fast transmission speeds, but the problem is you've got a lot of neurons in that head. And if every one of your axons was big enough to have that kind of very fast transmission speed, your head would be the size of this garage in order to pack all of those axons in there. So rather than doing that, vertebrates, things with backbones, came up with a different way to speed up action potential transmission. In things with backbones, there's this process called myelination. Myelination means wrapping this axon in bundles of a substance called myelin. So it ends up in a cross-section looking like that. If you were to take a cut through that and turn it so you're looking at the end of the axon, what you'd see is that's the axon itself, wrapped up with a cell which is wrapped around and around and around, kind of like a piece of electrical tape. And that cell is filled with the substance called myelin, which is sort of a fatty protein substance. This myelin serves a couple of purposes. First of all, by wrapping around this cell and sealing it off, we make it so that effectively there aren't any usable ion channels under there. So that if I have an action potential here, those ions that come in can't leak out in this part. They have nowhere to go, which helps to focus them onto this next area. It also means I can't have action potentials in that area. Remember, an action potential is a series of changes coming from opening and closing ion channels. That can't happen here. But since the ions can't leak out, it kind of helps focus this change on this next part. It also limits some kinds of electrical interactions between what's going on in the cell and what's going on outside in a way that helps focus the signal. So the action potential happening here, that change gets focused onto the next part that doesn't have myelin. We call these open sections, where there is no myelin, nodes of, now, hope you can see that, it's not lost in the glare. The word there, just in case, is this. It's the name of a person who discovered it. And I don't speak French well, so I usually just say it nodes of Ranvier. But I believe it's something like Ranvier. Anyway, but it's more fun to say Ranvier. So, the action potential that happens here effectively kind of focuses a change very quickly on the next node of Ranvier, so that this action potential triggers another action potential here, largely skipping the area in between. If you looked at this from the outside, you'd see action potential, action potential, action potential, as if it sort of jumped from one node to the next. And it takes very little time to get through this area. From the inside, you could see that there is an electrical change getting pushed to the next section, but it moves much faster. You don't have to regenerate that action potential at each part. You can't have these arbitrarily long. There is some signal loss under here, so that if you just myelinated the whole thing, the signal wouldn't get through. But by keeping them a little bit short, you can speed this up enormously. So you can have a small axon where our action potential goes blip, 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 blip from node to node, rather than on each little bit of membrane. Speeds it up a lot. Now that myelin, this cell that makes the myelin in the central nervous system, in the brain and spinal cord, this is sort of a pseudopod of one of those glial cells called an oligodendrocyte. It has a little piece that reaches out and wraps itself around another cell's axon, forming part of the myelin sheath. In the peripheral nervous system, each one of these is its own cell, called a Schwann cell. But same idea. The myelin is kind of grayish-white in terms of color, which is one of the differences between gray matter and white matter, if you've heard of that. We'll talk about this again in the next lecture, but white matter is called white matter because it looks kind of whitish, and that's because white matter is mostly myelinated axons. Gray matter is where you find the cell bodies and dendrites, which don't have myelin. So they aren't quite the same. They don't look quite the same. In any case, the name for this way of transmitting action potentials, which makes them much faster, is saltatory conduction. That word saltatory, the salta is from the same as the Latin root for jumping, to jump. And if you speak Spanish, saltar, to jump, you get the idea. All the Latin-derived languages have that same kind of root for that word. So this is jumping conduction. It goes boing, 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 boing from one node to the next. All right, so we've figured out how that action potential moves down the axon. Now, what happens when it gets to the end? That's our next question. So let me erase this. and take a look at what happens at the end of the axon. Actually, I'm going to draw that a little differently. So the big view is that I've got one axon, one neuron here with its axon, and then axon terminals onto other cells. And what I'm going to be looking at is this right here, where the axon terminal of the first cell comes up to some part of the second cell. So we'll draw that here. Here's our axon terminal, and here is the next cell. The name for this whole junction, this whole thing, is a synapse. So this, the axon terminal part, is the presynaptic cell. This is the postsynaptic cell. The two of them are not physically touching usually. Instead, there is a very small gap between them. There's several names for this. You might hear it called the synaptic cleft. I like synaptic gap. But there's many different names for it. It's very, very narrow. We're talking dozens of nanometers, I believe. I'll have to look it up to be sure. Now, let's talk about how this works. So, this here is an axon terminal, the end of the axon, and there's some specialized structures in an axon terminal. One of the most important ones for our purpose here are these things called synaptic vesicles, which are little bubbles of membrane that are filled with some kind of chemical. The generic name for that chemical is I'm going to erase this word in a second, neurotransmitter. So a neurotransmitter is any chemical used by an axon terminal, a presynaptic cell, to then send a signal to a postsynaptic cell. There are lots of different neurotransmitters, and we're going to talk about a few of them in a little bit here. I'm going to erase that word so it doesn't get in the way here. Now, one other thing of importance. On this presynaptic cell, I have voltage-gated calcium channels. We haven't really talked about calcium yet. It's another ion, calcium, Ca2+. This cell makes sure there is some calcium out here, but virtually none inside the cell. That's because calcium coming in is going to be an important signal. So just remember there's calcium out here, but really none in here most of the time. All right, over here on the postsynaptic cell, we're going to have some kind of receptor. Some kind of membrane protein that will allow this cell to respond to those neurotransmitters. Now, those could be chemically gated ion channels, so that this neurotransmitter will bind to them, they'll open, ions will flow in, and we'll get a graded potential, just like we talked about earlier. Or they could be G-protein coupled receptors, so that my neurotransmitter will bind to them, they will use a G-protein to influence usually an enzyme, that enzyme will make a second messenger, and that second messenger will cause an effect in the cell. Both of those are options in the nervous system. If they are ion channels, we call them ionotropic receptors. If they are G-protein coupled, we call them metabotropic receptors. Just some names we use for those things. All right, that's our scenario. That's the stage that we've set. So let's see how this works. I'm going to erase these words so I have a place to write the steps over here. Get rid of these just so we don't get too confused. I'll leave the calcium there. All right, so step one. the action potential propagates down the axon to the axon terminal. So here comes my action potential, propagating down here. Step 2. Depolarizing the terminal opens the calcium channels. So when that action potential gets here, the flood of sodium coming in changes the voltage here, which opens our calcium channels, which means calcium comes in. So in comes some calcium. I'm going to be a little vague on what exactly happens then. The calcium comes in, and what it does is activate some machinery that's sensitive to calcium here. I'm going to use the word Shazam here, which I use kind of as a symbol to mean stuff happens here that we understand pretty well, but it's very complicated and we really don't need to get into it in detail right now. So this is a Shazam type mechanism. So calcium comes in, Shazam! That leads to these vesicles moving up and docking with the membrane at the end of the axon terminal. So the vesicles dock and release their neurotransmitter. That neurotransmitter is now released into the synaptic gap. This is a very small space, which means very quickly this diffuses across, and some of it binds to receptors. NT, neurotransmitter, binds to the receptors, which causes some kind of effect in the postsynaptic cell. And that will depend entirely on what kind of receptor that is. If it's an ionotropic receptor, that means we're opening or sometimes closing an ion channel, which will cause a change in the membrane potential here. In other words, it'll cause a graded potential. If that's a metabotropic receptor, a G protein coupled receptor, it'll cause the creation of a second messenger, which will cause some other kind of change. And that can be complicated. It could be all sorts of things. It could be a slow change in membrane potential. It could be a change in the kind of things that are going on in that cell. It could, for example, if that's a glandular cell, it could cause the cell to start secreting or not secreting a hormone or some other messenger. There's lots of possibilities. And what happens depends entirely on what receptor that is. We'll get to this in a second. So, that's the general process of transmission, but there is an important step that we haven't talked about here, and that's the idea of termination. My action potential gets here, we release the neurotransmitter, it causes an effect, and then what? Because that all didn't take very long, milliseconds. When the next action potential comes down here, we don't want this, probably, to still be going on. So we need a way to end this, to stop releasing that neurotransmitter and stop the neurotransmitter from having an effect on these receptors. I poofed that neurotransmitter out here. Do I just let it stay forever? Probably not. So, I need some way of ending this, terminating the signal. There's two main ways of doing that. The first is that I can break down the neurotransmitter with an enzyme. Sometimes what we have is an enzyme here on the postsynaptic cell. And when the neurotransmitter binds to the enzyme, it gets broken down. So it's no longer a useful neurotransmitter. So imagine it might work something like this. Let's imagine that we release 10 molecules of neurotransmitter into this gap. It would actually be many more than that, but we'll just say 10. So let's say it has 50-50 chance of binding to a receptor or an enzyme. So we release it, half of it binds to receptors and causes an effect. The other half binds to enzymes and gets broken down. The stuff that's on the receptor doesn't bind very tightly, so it pops back off. So now there's 5 of them here in the gap. Round 2, half of it binds to receptors, half binds to enzymes. So it reshuffles and binds, and let's say 2 of them bind to enzymes, those get broken down. 3 of them bind to receptors, causing a continued effect. Those pop off, and now we shuffle them around. 2 of them bind to enzymes, get broken down. The 1 binds to the receptor, causes a little bit of last effect. That pops off, and then that probably binds to an enzyme, and then we're done. We broke down all of that stuff as it sort of shuffled around in the synaptic gap, preventing it from constantly causing an effect on the postsynaptic cell. The other main way that this can happen is reuptake. Sometimes what we have, instead of enzymes over here to break it down, sometimes what we have is transporters on the presynaptic cell to pull it back in. So in that case, it would be we release 10 molecules of neurotransmitter here. Half of it binds to the receptors, half binds to these transporters and gets taken back. Round next moment in time, that 5 reshuffles, 3 of it binds here, 2 binds here. Reshuffle, 1 of them binds here, 1 binds here, and so on. It still removes it. The stuff that gets taken back gets packaged back up into these vesicles and is ready to be reused. Depends on the neurotransmitter. Some neurotransmitters are broken down, some are recycled. There's 2 of them we'll use as examples for that. So, as examples of those 2 methods of termination, there's a neurotransmitter called acetylcholine, usually just often abbreviated ACH. Acetylcholine is usually broken down by an enzyme on the postsynaptic cell. That enzyme is, it's a good name, acetylcholinesterase. You gotta love some of the words you get in this class. They're just wonderful. So, acetylcholinesterase on this cell will break down acetylcholine so it doesn't stay forever. It'll allow us to terminate the signal. Another neurotransmitter, serotonin, that's an example of a reuptake type. There are serotonin transporters on the presynaptic cell that take serotonin back in. The reason I use these as examples is because there are pharmaceuticals and other chemicals which work by interfering with these, by affecting this termination process. So, for example, let's see, how am I going to write this? There's a nerve gas called serin. This enzyme is often abbreviated ACHE. Serin is an acetylcholinesterase inhibitor. I think that might be in the glare, so I think you've had enough time to look at this part of the diagram. So I'll write it again over here. The nerve gas serin is an acetylcholinesterase inhibitor, meaning it interferes with the activity of acetylcholinesterase. So in that case, think about what's going to happen. If serin is blocking this, I release acetylcholine, it binds to the receptors. It can't bind to the enzyme because that's blocked. So it binds to the receptors, causes an effect. Comes back off, reshuffle. Binds to the receptors, causes an effect. Comes back off, reshuffle. Still no enzyme available, so it binds to the receptors. It keeps causing an effect. if you receive a dose of this acetylcholinesterase inhibitor, when your presynaptic cell releases acetylcholine, it has a much stronger effect than it otherwise would. It keeps affecting the postsynaptic cell. That can be a problem because the neurons that control your skeletal muscles, the ones that you use to do things like move and breathe, use acetylcholine as their neurotransmitter. So if I am poisoned with sarin, a cholinesterase inhibitor, that means that when my motor neurons release acetylcholine onto my skeletal muscles, it stays more active in the synapse and the muscle has trouble stopping its contraction. So for example, if I take in a breath, the muscles contract to let me breathe in and then they lock up and I can suffocate. There are some medical cholinesterase inhibitors, not that many, but it's not just nerve gas. A lot of pesticides are also cholinesterase inhibitors, which is one of the reasons they are dangerous. So that's the idea of how that might work. Now, serotonin, there are serotonin reuptake inhibitors as well. So, for example, Prozac. We call that an SSRI, serotonin selective reuptake inhibitor. So rather than preventing enzymatic breakdown, Prozac means that when I release serotonin into a synapse, it binds to the receptor, and then since it at least slows down the rate at which it's being taken back up, it stays in the synapse and continues to affect the postsynaptic cell. Prozac is usually prescribed as an antidepressant. For reasons that are not yet entirely clear, some kinds of depression seem related to insufficient action of serotonin in the brain. And so a chemical which causes serotonin to have a stronger effect seems to help with some kinds of depression. It has many other side effects as well. So these are just a couple of examples of why messing around with the termination step of synaptic transmission can be important, both as a dangerous thing and as a therapeutic thing. And really, of course, those aren't entirely separate. So that covers most of what I wanted to talk to you about the basics of synaptic transmission. I did want to go through a few common neurotransmitters just so you recognize them and talk a little bit about their receptors. So some common neurotransmitters. Acetylcholine. There are two main groups of receptors for acetylcholine. There's the nicotinic receptors, which are sodium and potassium channels. So they are ionotropic receptors. There are also muscarinic receptors, which are G protein coupled. You'll learn about these when we talk about, you'll learn more about them later in the nervous system when we talk about the peripheral nervous system and in the muscular system. Your skeletal muscles use this kind of receptor. Some of your smooth muscle and some glands use this kind of receptor, so you'll see both of them. Some others that are involved. Glutamate is a common one. There are several kinds of receptors for glutamate. AMPA receptors are ionotropic. Sodium and potassium can pass through them, as well as some other ions. There's also NMDA type glutamate receptors, which are also ionotropic. But they have some really interesting characteristics. If the postsynaptic cell is depolarized, so for example, if you release glutamate onto an NMDA receptor, and it lets in some positive ions, and while that graded potential is still there, you activate that same NMDA receptor, it changes how strongly it will respond to the next signal. So NMDA receptors have this interesting characteristic where they change their responsiveness based on what else happens when they're stimulated, which kind of has some interesting implications for a nerve cell being able to start responding differently to an input if other inputs are happening at the same time. So that's kind of an interesting notion. And then there are several metabotropic types. Okay. GABA is a common neurotransmitter. The main kind of receptor for that, at least the one we're going to mention, is GABA-A, which is an ionotropic chloride channel. Chloride tends to go in and it tends to make the membrane potential come down. It actually tends to make it stay near resting membrane potential, interestingly. Which means that a GABA receptor, if you activate it, will tend to kind of keep the postsynaptic cell from becoming depolarized. We're going to talk about what that means in a moment here. And then we've got two more that I wanted to mention. Serotonin. The main kind of receptor for that are called 5-HT receptors. Most but not all 5-HT receptors are metabotropic. And finally, opioids. You have some what we call endogenous opioids, but a number of drugs of abuse also stimulate these. For example, heroin stimulates opioid receptors, and opioid receptors are all metabotropic. Now, let's take one more thing and talk a little bit about some of the terminology we use for postsynaptic responses, so in some of these cases. So you can pause this to write all this down, and then I'm going to erase it. So looking back really quickly at our synapse, if the neurotransmitter causes a change in it causes a change in membrane potential over here. If it causes a graded potential, so a postsynaptic graded potential, these can come in, we kind of have two terms we use for these. EPSP stands for excitatory postsynaptic potential. By excitatory, we mean that it's depolarizing. It brings the membrane up closer to where it would need to be to cause an action potential if it got to the hillock. Compare that to an IPSP, which stands for inhibitory postsynaptic potential. An IPSP is a hyperpolarizing graded potential, which actually, if that gets to the hillock, it makes it harder to bring it up to threshold. And for some students, that's a strange concept, because we think of the idea of one neuron sends a signal to another neuron to make it activated. But it's actually not that simple at all. It's just as common for one neuron to tell another neuron not to become active. Let's very briefly see why that might be of interest. We've spent a lot of time here on very detailed molecular stuff, so let's take one step up, kind of back, and look at some of why all of this matters. So let's imagine here's a multipolar neuron with its axon. And here are several more axons. Here's one that has a terminal there, one here, and one here. All right. Let's imagine this synapse causes EPSPs, depolarizing graded potentials, as does this one. So let's imagine what happens when cell A sends an action potential down here. Well, it'll cause release of a neurotransmitter and cause a depolarizing graded potential, which spreads out but decays, and before it gets to the axon terminal, decays too much. So, sorry, not the axon terminal, the axon hillock. So this cell makes a graded potential, but it's not strong enough to cause an action potential here. Too bad. Now let's look at cell B. Cell B does the same kind of thing. Causes a depolarizing graded potential. But again, it's not strong enough on its own. That ripple decays too far and does not bring this up to threshold. So a signal from cell B also does not cause the cell to have an action potential. But what if cells A and B receive signals at the same time? So cell A starts its graded potential, cell B starts its graded potential. And remember, when two graded potentials meet, they add together. So even though each one of them on its own is not strong enough to cause an action potential, where they meet, they add together into a bigger signal. And that one does bring the axon hillock up to threshold. It's like this one causes a weak signal and that one causes a weak signal, but together they're enough to trigger this. So that means this cell will send an action potential not when cell A sends a signal, or when cell B sends a signal, but only when A and B send a signal at the same time, which means this cell detects simultaneous activity in cells A and B. Now we've got some interesting computation going on. Okay, so how can we make this even more interesting? Let's say cell C causes inhibitory postsynaptic responses, causes a hyperpolarizing graded potential. Maybe it opens potassium channels, which let potassium out, which brings the membrane potential down, or chloride channels, which lets chloride in, which tends to hold the membrane potential at rest. Now notice it's a lot closer to the hillock. So A and B, let's say A and B both have their signal, so they both activate and we're having this signal coming in together and it's strong. And right before it gets there, a signal comes in on cell C, which is very close, so the signal is pretty strong still, and says, nope, it's a hyperpolarizing graded potential, and when it adds with that depolarizing graded potential, it neutralizes it, at least partially, and prevents it from causing an action potential. So now this cell says, I will fire if A and B have a signal at the same time, unless C is active, in which case it will keep me from firing. So now this is not just a simple, I will fire when that cell fires. This is saying, I will be active when this and this, but not that, are active. That's a pretty interesting bit of computation. Now, to give you a sense of the complexity of your brain, this is one neuron with 3 inputs. Your brain contains something on the scale of 80 billion neurons, each of which can have anywhere from hundreds to thousands of inputs. So the amount of computation that happens at any individual neuron, you could imagine if you've got 1,000 inputs here, each of those can be having graded potential several times per second. So those signals are spreading out over this cell and adding together and neutralizing each other, and the membrane potential here must kind of be going like this all the time. And all of that is going into saying, do we send a signal here or not? It's sort of a filter, computing based on what kind of inputs you have and where they come in. And then this axon feeds onto other cells, including maybe these, and maybe even feeding back onto itself. That happens a lot. So what you get here is a picture of the complexity here. People sometimes wonder, how could this lump of tissue, this brain that's three-ish pounds, make something like a mind? And what I'm telling you is, the complexity of stuff going on in the brain is enormous. More in many ways than any computer we have right now, although people are working on computers that could simulate that kind of function. And that will be interesting if we come up with computers that can function at the same level of complexity as the human brain. What happens if they act like a human brain? That kind of stuff is very interesting to me. We can talk about that in the Zoom session sometime if you want. But anyway, what I wanted to get you there is, why have we been learning all this? Well, for one thing, to show you a little bit about how this nervous system is going to work. Now, the next topic we're going to talk about in the next section is the idea of sensory information. How do we get information into this thing? How does a sensory neuron work? What is a sense? That's one of my favorite things in the entire class. So I'm really looking forward to talking to you about that, and I'll see you soon.